the first thing we want to talk about is revisit the cell membrane. So this is just another depiction of the cell membrane. I just wanted to talk about the biphospholipid layers here, and you can see the proteins that are embedded in here. And remember, they have the glycocalyx on the outside that can include some carbohydrate kind of change, something called a glycoprotein, which would be a carbohydrate attached to a protein. You can also have things like cholesterol in the membrane, which adds to the flexibility of the membrane. So it's good to have cholesterol, especially high density ones. And then you can see the cytoskeleton on the inside of the membrane. So that's just to revisit what the membrane is like. And of course we have these phospholipid bilayer. Whoops, I didn't mean to put that in. Phospholipid bilayers, and this is the model that we usually use. This is called the fluid mosaic model. One of the reasons why it's called that is because these phospholipids, remember, are not bound to each other. In other words, they're not covalently bonded to each other. Instead, they are just there because of their position in relation to water. Because there's water on the outside, or another cell that has water in it on the outside, and water on the inside, and that will make the heads orient outward because they are hydrophilic, and the tails orient inward. That means that they can flow past each other, and this is one of the reasons why you can pinch off a membrane part and make vesicles and lysosomes and so forth that we were talking about before. All right, so anyway, that's just to revisit the membrane a little bit. Okay, so we're going to talk about the different types of movement across the membrane, and we're going to start in this little section with simple diffusion. Now, simple diffusion is also called passive diffusion. Let me back up just a little bit. Also called passive diffusion is the movement of a small or uncharged substance down a gradient, or that is with a gradient from high to low concentration through the phospholipid bilayer, and it requires no energy. Examples of things that do this are carbon dioxide and oxygen, and they have to be small and uncharged because remember that if they were charged, they'd be repelled by that phospholipid fatty acid tails. And if they were charged positively, they would be attracted to the outside layers. So they couldn't easily travel across the membrane. Also, they have to be small to be able to fit between the spaces between the phospholipids. So this is a very fast way of things getting in and out of cells. But also there's not that many molecules that can actually do it, all right? Because many things in biology are going to have a charge of some sort. A perfect example of that is water, which is the next type of movement, which we call osmosis. Osmosis is the movement of water down a gradient or with a gradient, so that's pretty much the same, except for we're just talking water now. And it has to go through an aquaporin. This also doesn't require energy. Now, what an aquaporin is, and I've drawn it in this little diagram here, aquaporin is a protein that is a kind of a narrow tube, basically, that allows water, which is a small molecule, but it has the polar nature, remember? So it cannot then pass through this layer because of the phospholipid fatty acid chains, and the aquaporin allows the water to go through. This type of movement is also really fast. You can see it happen before your eyes, but it does require that there is this little port of entry. Now, it turns out there's a whole lot of those in a phospholipid bilayer, or that is the cell membrane, and so this happens relatively quickly in most cells. Okay, so there are a bunch of situations, though, that this is the thing that is super confusing, all right? So I am going to back out, take a second to write everything that I have right here down. So stop the video, write everything down, and then we're going to talk about it. So I'm going to start talking about it now, but you guys have taken a chance to write it down. Okay, so let's just talk about the first situation. Okay, most cells, and this is true for all the situations, but most cells are about 1% solute. And they will be either next to another cell, which is also 1%, solute. And of course, in between the cells is either middle lamella or glycocalyx, which is watery. And that's interstitial fluid is also usually about 1%. So really inside the cell or the animal or plant or fungi or whatever multicellular organism, it will be about 1% solute, okay? So the outside solution we're talking about is like a beaker of water for, just for example, or a lake or something like that. All right, so let's just say that the outside water situation is 100% water. There are no solutes dissolved in it. Now, in nature, this is uncommon. But in the lab, it can be fairly common. You can use distilled water and put a cell or a group of cells into it. All right. So in this situation, there's names given to the sides. So in this case, we have a situation where we have 1% solute inside, 0% solutes outside. So there's more water outside than inside, right? Because 1% is going to be 99% water inside. Okay, so the water is going to move from high to low concentration, and what's going to happen to this cell is it will expand. It's an animal cell, so it doesn't have any restrictions on it, so eventually it'll blow up, or what we call lyse. All right. Now, in this, a lot of times and in your book, this is what they say, this is a hypotonic situation. But you can talk about the cell as well. So in this case, the cell is hypertonic. So hypertonic means where the solutes are, and hypotonic means where the solutes are not, or that is where the water is. Okay? So always in a situation where you have a solution compared to a cell or some other body of waters, another solution, there is one side that's going to be hypo and one side that is going to be hyper if there's any movement of water happening. So you have to specify which side you're talking about when you use these terms. So for example, I couldn't just say this is hypotonic because I'm saying what's hypotonic, the solution or the cell. Okay. Now, if I were looking at a plant cell actually likes this situation. It will not lyse because it has the cell wall. Now remember, the cell wall is somewhat rigid, and it's rigid enough in this case to stop the expansion of the cell membrane. And so what will happen is water will move into it until the pressure from the cell wall pushes it back. This is kind of like if you were blowing up an inner tube. You're going to get to a point where you can't blow any more air in. And at that point, the inner tube is pushing back on your effort to blow in. And that makes it so that it's completely filled. And in this case, in a plant cell, when it's completely filled, it's called turgid. All right, so plant cells like this because it makes them, all their cells really tight, and then they can press up against the neighboring cell very tightly, and the communication is much better, and the plant stands more upright, etc. So plants actually really like this situation. So they like to be in hypotonic solutions where the inside of the cell is hypertonic. All right, now the next situation is the opposite. Okay, so in this case, I'm placing it in salt solution. So I'm placing this animal cell in salt solution. The solution itself is hypertonic because it has 10% solutes. That would mean it has 90% water. Compared to inside 1% solutes or 99% water, the water will move out. Now, that is, animal cells, will shrink and they will become raisin-esque. That is, this red line here would be what it would look like after it shrunk. And that is called crenates. All right, so this would not be a good situation for animal cells either because they would eventually, the solute concentration would increase, all the organelles would be squished down, and they would eventually, it could stop if there was an equilibrium reached, but mostly it's probably going to be dead because it's going to have no water inside of it. All right, so in this situation, then this is hypertonic on the water side, or on the solution side, and hypotonic on the cell side. Now, plant cells will also do that. In this case, though, what you see is the cell wall will remain and the cell membrane will crenate inside. So they give it a special name because it's the plasma membrane is pulling away from the cell wall, and it has a special look to it, so they call that plasmolysis. All right. Now, lastly, we have a situation that is isotonic. All right. Isotonic means that there's the same amount of solutes on both sides. All right. So the solution has 1%, the cell has 1%, and the water will move back and forth. If one water molecule moves one direction, then the concentration slightly increased, and another one will move the other direction. This is what we call a dynamic equilibrium. This is isotonic. Both the solution and the cell would be called isotonic in that situation. So animal cells love this, all right? And this is where they would like to be, which is why in a multicellular organism, everything's roughly the same, okay? I mean, you know, in general, roughly the same. In plant cells, they don't particularly like this. This is not bad for them, but it's not the best, all right? So animals like this the best, plants like this the best.